Alright, let's go see this process in action, because it is really complex. So let's start out with the ribosome, just so we have all the parts. The ribosome has a small subunit and a large subunit, and two pockets. This is where the tRNA comes in. There's going to be a P site and an A site. The A site is going to be where we're going to get started. The P site is where we're going to make a type of bond called a peptide bond, and that's going to keep the chain of amino acids growing. So you could see at the ribosome, this is where all of the different pieces come together. Number one, that thing that looks like a hamburger bun in the back, those are the two pieces of the RNA or ribosome. We have the mRNA, that's going to be the red or purple area. That's the code that's going to build our amino acids. Remember how we read those in three-letter codes called codons? That's what you're seeing there. The green things, there are two tRNA. One's in the P site, one's in the A site. The arrow's on the one in the A site. The bottom part is an anticodon. I think that'll make a little more sense in a second. Finally, this will be the amino acid that's going to use a special type of bond called a peptide bond to bind that chain of amino acids together to get longer and longer. And growing peptide chain is also the amino acids that are going to be in that primary phase of elongation. So let's start with the translation process. Initiation, just to kind of give a quick recap, is where mRNA binds to the ribosome subunit. A large ribosome unit will sort of come together, and we're going to start transcribing the mRNA at an AUG site. Elongation is where we're going to start making a longer amino acid chain. And then finally, termination is kind of like that period where we terminate the process, the protein is now completed, and it's going to get just packaged and sent wherever it needs to go. Or usually what happens is it'll get added with other proteins to be an active protein or enzyme or hormone or whatever it's making. So here's the first process. So we're going to say that the mRNA came out of the nucleus and has now come to a ribosome. This is going to be in the rough ER. We're then going to go ahead and have the mRNA come together, and it's going to sort of feed in almost like a tape. If you remember those old tapes, tape cassettes or something like that, where it had a little bit of that reel before it actually started playing the music or whatever. And we know that our first site is going to be AUG, which is also known as our start codon. And we're going to go ahead and get that process started. So we have our start codon, and our first amino acid is going to come together. And if you look at that table that we looked at, our first amino or codon, AUG, will code for a amino acid called methionine. And that's what you see here. We just wrote met, just so it would be faster. That's going to get the other part of the ribosome to initiate. And now we're ready to start making that protein. So the large unit has come on. That's the P site, and the A site is now available. We're going to do this in two steps. First step is mRNA binds to the small subunit and delivers the methionine or the start codon. The second unit is a large subunit, and that's going to be in the P site. That's going to be in the P site. And then we have A site open up. So let's see this in action. So the steps that you're going to see is that number one, the codon gets recognized by the tRNA, and it's going to bring an amino acid, the appropriate amino acid, to initiate the process. There's going to be a peptide bond. It's a bond that forms between two amino acids that will be formed, and it comes through dehydration reactions. Remember where we remove the water to make something bigger. Finally, we're going to do translocation where the tRNA is going to be removed, and we're going to just keep doing this over and over again. And this is going to require some energy to get this process started. So I've made it upside down, but I've typed in all the numbers so we can kind of see what's going on. So number 1, you'll see that we have our P site and our A site, and that this is our mRNA. Our first code was AUG, and that tRNA came and brought methionine. Our next code was GGA, and if you go to that table and look up GGA, what you'll find is that that will be the amino acid glycine. The thing that I did want to show you is, remember how on the tRNA we had this thing called an anticodon? That's what this dark green color is. Anticodon is the opposite of what's on the mRNA. That's how it knows it's bringing the right amino acid. So for instance, the opposite of AUG is going to be UAC. So if this is the codon GGA, the anticodon would be CCU. Well, let's see what happens next. What is that bond called? Do you remember? It's called a peptide bond. And then what is the process that causes this bond called? It's just the peptide bond formation. We're then going to go ahead and have our next tRNA come, and how we know that this is the appropriate tRNA, it's kind of like our double check, is that UGU, the anticodon for that should be ACA. And that bonds through. We're going to have the shift occur where that first one leaves the P site. Can you see how that tRNA left? Can you also see how it left the mRNA behind? It's now going to go float in the cytoplasm, find another methionine, and then just wait. And if it ever sees another site like AUG again, it's going to come and drop it off. But it doesn't look like it's going to be needed for a while. So the next thing that happens is we have our next codon that looks like it's about to come, AAG. And the anticodon for that would be UC. So we're going to see a shift occur again. Remember how this will have to come into the ribosome. This will have to get shifted out and leave, and it's going to leave its glycine. And that's what's happened. And keep in mind, just for my little picture that I made, I didn't want to sit here and do thousands of them. Really, a small protein would have a couple thousand amino acids. But just for the sake of, us doing this little exercise, I just made it so there's 4 amino acids. And finally, we're going to have our stop codon. Here's another one, CGA, anticodon is GCA. Another thing you can see now that everything's shifted over. This is the P site, this is the A site. The tRNA floated away, but you can see the chain is still connected by those peptide bonds. Finally, we hit termination. And remember those stop codons in the table? That's what we're going to do, and we're going to terminate this process. The mRNA is a stop codon. There's no amino acids really brought by this process. The small and big subunit just separate, and that's going to make sure that the amino acids just drift to wherever they need to go. The mRNA is released, and then the completed peptide chain or amino acids are now going to do whatever they need to do, and it's usually to go to the smooth ER. So what happens with UAA, and if you look at that table, you'll find out UAA is a stop codon. It brings this thing called a releasing factor. When the releasing factor comes in, you can see everything goes apart. The mRNA is gone, drifts out. The ribosomes separate out, and then the protein is going to go into the smooth ER, and then eventually get packaged a little more, maybe do some folding, and then finally go maybe to the Golgi apparatus. So, big recap. DNA to DNA is the nucleotides. We discussed in one of our first videos was DNA to RNA, and that was called transcription. That had that little editing step that occurs first before it leaves the nucleus. And then outside the nucleus in the cytoplasm, we do that thing called translation. Great.